We would deal with role of neutralizing antibodies in COVID-19 infection and vaccination. As you have seen in my life, I've tried to cram a lot of things, but in this presentation, we will stick only to neutralizing antibodies. A number of questions were shared with me from the participants, and I would think that by the end of the presentation, at least answers to 7 of them would become clear. We intend to hold a webinar along with Principal Faridi of ERA Medical College on MIS for children, and I think we would discuss the multi-organ inflammatory syndrome in greater detail then. We know that SARS CoV-2 is a novel RNA virus. This has a nucleocapsid which is consisting of RNA. With this nucleocapsid protein is the most abundant and immunogenic of the antigens present. But as you see here, this is enclosed inside the virus. So antibodies to this will have no protective role. On the other hand, the virus is covered over with glycoprotein known as spike proteins. And it is this spike protein which binds itself to ACE2 receptors which are present on the respiratory secretion and other cells of the body before it gains entry.
The large S glycoproteins are used by the virus to gain entry to human cells. They most likely attach to angiotensin converting enzyme 2 receptors on the cell membrane allowing the virus entry.
Could you please voice over, sir, because it is stuck in voice is not clear.
So it becomes clear that spike protein plays a very important role. Now, papers from China had indicated that a person responds by producing antibodies.
Sir, voice में vibration है.
These antibodies appear to be of 2 different types. One, for those who have
sir, you can mute your video, so voice will be clear then.
Video is muted.
This
is now slides only.
Yeah. Okay, now it's clear. Now it's clear. Yeah.
So that the response to the viral infection could be divided into 2. Those who suffer from severe and critical infection produce a large quantity of antibodies, while majority of patients who have mild infection have very few antibodies. But you would notice that persons who are likely to donate convalescent plasma are those who suffer from mild infection, while those who are admitted and critically ill are less likely to donate their plasma, though they have more antibodies than those who have suffered from mild infection. And because we were not monitoring presence of neutralizing antibodies when we were doing a convalescent plasma trial, the plasma trial failed because we did not know what was the quantity of antibodies present. But there is protection after infection was very clear early on with animal experiments, where after the animal had recovered it was not possible to reinfect the animal. It was also shown that if you take plasma from a protected animal, then and you inject it into Syrian hamsters who were the animal model, then it is not possible to infect Syrian hamsters with SARS CoV-2. You would also remember that experimental monoclonal antibodies were used in then President Donald Trump when he was reported as RT-PCR positive for COVID, and within a few days he was back in office and campaigning. There's also been report that if the donors had anti-spike antibodies of more than 1,000, then if it is given within 72 hours to persons who are not vaccinated, then it will save them from progressing to severe disease. You would also notice that most of the vaccines are focusing on spike protein. So antibodies to spike protein are of paramount importance. Of course, in the last column, you will notice that they also looked at cell-mediated immunity. And though we are not measuring cell-mediated immunity at this moment, it appears that it does play a role. But what we can quantify are the antibodies. Once monoclonal antibodies became available and we could understand how the ACE2 receptor and the spike protein interact with each other, they looked at the epitopes on the spike protein, and they found that there are at least 18 to 19 epitopes, which if you block, you can cause neutralization. The whole sequence of spike protein consists of 1,273 amino acids. And this has two portions, S1 subunit and S2 subunit. In S1 subunit, there is a region which varies from 319 to 541, which is known as receptor binding domain. So it is not the whole spike protein that binds, but it is receptor binding domain which binds to the ACE2 receptor. And even in the receptor binding domain, there is a receptor binding motif which varies from 437 to 508 amino acids. It is the receptor binding motif which actually binds to ACE2 receptor. Now, because it's an RNA virus, there is a lot of mutations that occur. And though it has a RNA repair mechanism, but still it accumulates 1 to 2 mutations every month. This is half of what influenza virus accumulates and 1/4 of what HIV virus accumulates. So coronavirus, SARS-CoV-2 mutates just as every RNA virus does, but at a lesser frequency than influenza and HIV, which are other important RNA viruses. But you will notice that wherever there is mutation in the receptor binding domain, then you have problem. And the Delta variant that is there is because there have been 2 mutations in the receptor binding domain. In the whole virus, there seem to be at least 16 to 17 mutations, but it is the receptor binding domain which really matters, and the mutations in receptor binding domain really matter. This is how the British uh, health authorities indicate that the Delta virus must have P681R mutation, while Beta must have K417 mutation, Gamma must have K417T mutation, and the South African mutations had E484K. They have seen that Delta virus that was responsible for the second wave in India had increased transmissibility as compared to the wild strain. It had more infection, and infection spread rapidly in household contacts. The immunity as a consequence of vaccines which use the wild spike gene was compromised to a certain extent, and even those who had had infection in the past last year were still susceptible to fresh infection. Last year, there was one change, which was D614 from the Wuhan virus. There was one change where D aspartic acid changed to glycine at position 614. And this gave advantage in the fit as a consequence, as you see there, that initially in February 2020, the virus was less than 1%. And by June, nearly 90% of the viruses were of this DUG mutation, D614G. Now in India, we've not been sequencing as much as we should. In England, with they sequence at least 5% of the isolates, they notice that since May of this year, Delta variant has become the dominant variant which is available as causing infection. And it is not only in India that we've had Delta variant, but in Singapore, in Indonesia, in Russia, in Africa, and in USA too, from 5% to 10%, now 20%, it's rapidly spreading. To make diagnosis using serology, for diagnosis, as you know, RT-PCR is the method of choice. And we have another seminar on the 9th where we will deal with that it is wrong to use RT-PCR for everything. And the title of that talk is One Size Does Not Fit All. But today we concentrate on serology. In serology, if the antigen used is nucleocapsid protein, since the antigen is within the cell, within the virus, it will only indicate whether a person has been exposed to the virus or not. So for epidemiology, you can use an antigen which looks at nucleocapsid protein. If you want to study whether a person has protection, then spike protein, because that is where it uses spike protein to bind to ACE2 receptor and cause and enter into the cell, is important. And to be more specific, rather than the entire spike protein, which is 1273 amino acid long, it is to study the interaction with our receptor binding motif inside receptor binding domain. If you block the receptor binding motif, then the spike protein will not be able to bind to ACE2 receptor and you'll have neutralization. Now, the antibodies that can be detected are either total, that means you don't differentiate between IgM, IgG, and IgA, or IgG. And neutralizing antibodies are normally of IgG type. Then the assays could be binding assay, where you study whether the antibody has bound to the assay, or functional assay, which is neutralizing assay. Neutralizing assays are functional type, which indicate whether this is how the virus works and you are neutralizing the activity of the virus. So as I was trying to indicate that not all antibody tests are equal. One is about the sensitivity. You would recall that in March or April last year, ICMR had permitted lateral flow antibody test to be used for diagnosis, and it was a miserable failure. As a consequence, they withdrew it. Of course, it was a wrong decision, and that is not where antibody tests, and especially a crude test like lateral flow, to be used. But if you want to study whether a person has been exposed to the virus or not, then studying detecting total antibodies to nucleocapsid protein could be used for epidemiology to find, because the virus is novel. So most of us do not have any antibodies to it. So when we have any antibody, that means we have had a subclinical infection with the virus. So that is the appropriate time to use nucleocapsid protein. For binding to spike protein, this will be surrogate for protection. Surrogate for protection is binding antibodies to ACE2 binding antibodies to spike protein. Now, since nucleocapsid protein is inside, it will be clear that antibodies to them are of not protective nature. That would mean that there will be antibodies which bind to nucleocapsid protein of coronavirus, which provide no protection. On the other hand, if antibodies will bind to different sites of S2 receptors, it will be binding antibodies, and it will show you whether there is surrogate for protection. But one example of protection is that if a person with knife enters into your room, what you want to do is hold the arm, catch him by the arm, by the hand which is holding the knife. This way you will neutralize his attack. On the other hand, if you bind to other portions of the body, that will have binding effect, that will block him, but it may not give you full protection. So binding to spike protein does provide protection, but it does not mimic real life situation. So you should imagine that there is ACE2 receptors on the surface of human cells in the respiratory tract, and the virus comes near it and attaches itself to the ACE2 receptor through spike protein. Your aim is that when the virus comes to the comes near the human cell, antibodies will block will attach itself to RBD or RBM, receptor binding motif, and prevent the entry of virus into human cells, since it cannot attach itself to ACE2 receptor. The test actually is done on viru cells. There are cells grown on cell culture, and then the respiratory secretion is poured over that. The virus will enter, and it will cause, once it keeps on multiplying, it will cause cytopathogenic effect. The holes that you see here are as a consequence of a cytopathogenic effect on viru cells. If the secretion was mixed with the plasma, allowed to neutralize for some time, and then poured, you would find that if neutralizing antibodies are present, then the cytopathogenic effect will be blocked. And this shows neutralization of the virus. But the trouble is that since the virus is highly infectious, this neutralizing test can only be done if you have biosafety level 3 laboratories, which India has in National Institute of Virology in Pune or NCDC Delhi or Jalma in Agra. There are a few national BSL-3 labs, but there are no private labs which have BSL-3 facilities. And besides BSL-3 facility, you would take 3 to 5 days for the cytopathogenic effect to appear and for the results to come back. So scientists in America as well as in China made a virus where the DNA, where the gene of spike protein was used to, to be inserted into ordinary virus, which is non-pathogenic. So when you insert the spike protein gene into a virus, this becomes a pseudovirus. Pseudovirus, and because the spike proteins are that of the coronavirus, so the specificity of neutralization would remain, but the potential to cause harm is removed. So the pseudovirus was developed, and it was first established in China, which was then published in Nature, and a simplified version was made available. The net result was that it is possible to perform this test using pseudovirus in clinical labs, which have BSL-2 facilities, but it will still take 3 to 5 days. And since time is of critical importance, the next best thing, the third approach, was to genetically engineer receptor binding domain and ACE2 receptor. So ELISA plates will be coated by ACE2 receptors, indicating or mimicking what really would happen in the respiratory secretion. And then there is RBD and plasma, human plasma or serum, are incubated in a tube separately, hoping that RBD, which is linked to an enzyme, will be blocked by the neutralizing antibodies, and then you will pour them over ACE2 receptor. After some time, you will wash them so that only the RBD, which has not been neutralized, would be able to bind and will be able to produce color. So the development of color is inversely proportionate to the neutralization power of the plasma or serum under test.
SARS CoV 2 is the virus that causes COVID-19. These antibodies, termed as neutralizing antibodies, stop SARS CoV 2 from infecting the cell. Not all antibodies that target SARS CoV 2 can stop an infection. Other antibodies
The voice is breaking this video.
This test was used in China in persons who had COVID infection and controls. And as you noticed, that high levels of correlation was found both in China and in Singapore when it was tested in COVID persons. Subsequently, it was given approval by EUA, emergency use authorization was provided by US FDA, and it's also been provided by ICMR and DCGI, and it's been made available. And when you do the result, then you find that the international available, which is costly, and the new test which has been introduced by J. Mitra have very good correlation. And the R2 is 0.9768, which is, that means you can use these tests interchangeably and you'll get the same results. We did, and we had been doing antibody testing. As I said, if you do total antibodies will only indicate whether a person has been exposed, and only 552, that means about 20% of those that we tested came positive. But if you look at protection, you would find that majority of persons had low titer antibodies. Most of the spike protein antibodies were low titer. This was, of course, done last year. Then, and as I've tried to indicate, that the presence of different concentration of spike antibodies are correlating with neutralizing antibodies to a certain extent. It's not as good as CPAS and J-Mitra's neutralization test, but you notice that some correlation is present. When we got this kit, we thought we should do the test ourselves. In the last 2 weeks or so, we have taken sample from 35 of our staff members. They vary from age of 21 to 69. 23 were male and 10 were females. 20 were vaccinated, 3 partially vaccinated, and 10 non-vaccinated. Among those who got vaccination, 9 were not infected, but 11 were infected in the Delta wave, second wave that we have. 1 dose and not infected were 3, not vaccinated and not infected were 8, and 2 were not vaccinated and infected. When we looked at the antibodies, we noticed that after vaccination, IgG titer of more than 100 AU was seen, and neutralizing antibody was more than 30%, and low and neutralizing antibody was present in 2. But if a person has had vaccination as well as infection by the new during the second wave, then high titers of antibodies as well as neutralization going on to 99.23%, 99.59%, 98, 99, as you notice there, very good neutralization is seen. Of course, I am one of the persons who do not have neutralizing antibodies. And the correlation between anti-spike and neutralizing antibodies is slightly loose rather than tight, so that anti-spike would not be able to address neutralizing antibodies at lower titer. At a higher titer, when anti-spike is more than 400, then neutralizing is also about 99 or above, and about 18 of them had correlation. But as you notice here, at a lower level, a lower titer, the correlation is not very tight. When you review the literature, which was published in Lancet for Covishield, they had mentioned that if you increase the duration between doses, then after 12 weeks, from a titer of 60, the titer will become 63,181. But this was the condition when you use the Wuhan strain, and the infection was by the Wuhan strain. So this was what was published in November. So right in November, we knew that giving 1 dose was good, and increasing the distance between 2 doses from, rather than 28, if you give after 28 days, then you found that the dose became 80, the second dose will produce 8,000. But if you gave after 12 weeks, it was 63,000. So we knew that if you increase the dose, increase the duration between the first and the second dose, the antibody titer will be very high and very good. It's also been reported from New York that the difference between seropositive and seronegative after the first dose itself was tremendous. Those who were seropositive would react very well after one dose. So maybe those who are seropositive, which were infected last year, one dose would work very well for them, and the second dose does not add much more to antibody titer. But the side effects, the systemic side effects increase. This was also seen with healthcare workers, those who had suffered from asymptomatic infection. When they got one dose, they produced high level of antibodies, and giving a second dose did not improve the situation. So those who have been infected once, maybe one dose is enough for them. It's also seen that the property of antibody production is age dependent, and they had noticed that when the infection, when it was given in elderly, those who were less than 60 responded very well after the second dose, but 31% of those over 80 years responded not as well. As you notice here, it was both for neutralizing antibodies as, and you notice that persons who were older had less antibodies, and some of them, like me, as well as Dr. K.K. Agarwal, did not produce antibodies after the second dose also. Pfizer then looked at when the variant emerged, then they investigated the variant's ability to escape, immune escape from those, from serum which were collected from persons who have been vaccinated. And the original N501Y strain did not have any immune escape. But Moderna had indicated that B1351, which is the strain, which was the variant observed in South Africa, which had E484K, that had some immune escape. This is an important study which has recently been published in June 28 about AstraZeneca, that is Covishield. They have used in London, they have used live SARS CoV to isolates to determine neutralizing antibodies in 106 vaccine recipients. After one, some of them were after one dose and others were after two doses. SARS vaccines, the strains which were used was Wuhan strain, that is the wild type, then the D614G, this caused the initial outbreak in 2020. Then they had the alpha variant, which is B117, or the beta variant, which is B351, and the delta variant 167, which is the strain, the variant found in India. So they used the wild strain, the initial UK strain, then the Nelly mutation, that's the alpha, beta, and delta. And they found that 2 doses of vaccine generated neutralizing antibodies against wild type, which had a neutralizing antibody of 419, which was 2 times less than what was being produced after Moderna vaccine. So their conclusion was that Covishield is at least 2 times less immunogenic than Moderna vaccine. This neutralizing antibodies were further reduced when you tested against either the alpha strain or the beta strain and the delta strain. And 60% loss of activity would occur against the delta. While Moderna will produce antibodies, 95% of all participants, the 106 had produced neutralizing antibodies against all variants. So it would indicate that Covishield, there is some loss of neutralizing activity against the delta Delta variant if you have been inoculated with Covishield. They also concluded that vaccine type was associated with decreased neutralizing antibodies, which was independent of SARS CoV-2 strain after 2 doses of Covishield vaccination. If prior infected persons with COVID symptoms, they had 32% higher neutralizing antibodies even after a single dose. This is what I was showing earlier. After a single dose of vaccination, all participants had neutralizing antibodies to the Wuhan wild type, but it had 65% less against the UK alpha variant or 88% less against the beta variant and 85% less against the delta variant. Previous history of COVID-19 symptoms was associated with increased neutralizing antibodies independent of SARS-CoV-2 strain after single dose of vaccine. Previously, they had already reported that elderly persons produce less antibodies, and they indicate that 2 doses offer better protection. Further booster may be required, and I think the CEO of Pfizer is indicating that a booster may be required after 1 year. Prevention of infection requires higher neutralizing antibodies than prevention of severe COVID-19 disease or death. And it was also seen in the recent outbreak, second wave, that persons who were vaccinated did not normally die, except if they did not produce neutralizing antibodies. So before I conclude, I would like to also mention that though SARS variant of concern partially escape humoral, but they do not escape T cell response in convalescent donor donor serum, and Euroimmune from Germany as well as Kaizen has launched T cell response kits, but at this moment we still do not know what should be the titer that would be indicative of safety. So that would mean that CMI cannot be ruled out, but at the moment 5 or 6 monoclonal antibodies have been approved by US FDA, and there is evidence that if you give a combination, a cocktail of antibodies which react with different sites in the receptor binding motif, then the combined version, if given within 72 hours to persons who are RT-PCR positive, but have comorbidities, so they might progress to severe infection, then you would save their lives. So antibodies would work if it is given within 72 hours of a person showing symptoms of COVID with comorbidities. What level is required? That experiment was done with rhesus monkeys, where they injected different concentration of antibodies to groups of monkeys and then infected them. And then only one set, which had received 250 milligrams per kg body weight, were protected, fully protected. And when they were fully protected, they had anti-spike IgG antibodies of 400, RBD ELISA titer of 100, or neutralizing antibody titers of more than 50. In our kit, we are saying that if it is more than 30% protection, then 30% inhibition, then you are protected. So in conclusion, it is there that our aim is to prevent this. The neutralizing antibodies will bind to RBD and prevent the attachment of spike protein to ACE2 receptors, so they will not be able to enter inside. To understand why some persons die and others do not, there was this study published in Nature Medicine, which indicated that persons who expired do not produce neutralizing antibodies. And it's been seen, this is from Yale, that mortality in COVID-19 correlates with delayed kinetics of neutralizing antibodies. Normally, neutralizing antibodies appear by the 9th day of infection. So if by 14th day antibodies have not appeared, then that might lead to fatality. We know that the COVID infection has 3 phases. First is viral phase, then is pneumonia phase, and finally is inflammatory phase. You should think of infection as only the virus is pulling the trigger. The injury is caused by cytokine storm, which is exaggerated response of individuals. So the damage is done by individuals, the trigger is pulled by the virus. And it is the presence of neutralizing antibodies on day 14 that will indicate that the person will survive or will not survive. So when should the test be done? After vaccination, after at least 2 weeks of second dose, you are looking for more than 30% of inhibition, which is adequate for protection. After infection, by day 14, if there are no neutralizing antibodies, this might lead to problem. And after infection, if planning to use monoclonal antibodies, immediately when RT-PCR is positive, you test for the presence of monoclonal antibodies. If they are absent, then monoclonal antibodies will be life-saving. If they are present, then monoclonal antibodies are not required. In India, our deviation from COVID-appropriate behavior is currently the biggest variant of concern. I thank you for your attention and hope that you stay positive but test negative by RT-PCR and have protective neutralizing antibodies after vaccination.